Microbial ecology focuses on the ways that microorganisms deal with or adapt to such factors as heat or cold, gases, acidity, radiation, osmotic and hydrostatic pressures, as well as even other microbes. These environmental factors affect the functioning of enzymes, and the survival of an organism depends on whether the enzyme systems can really adapt to the alterations in habitat. Sometimes we'll see growth, and other times we'll see existence or tolerance without growth. Note the difference, because growth means that they can grow and reproduce. Tolerance means that soon they might actually die. Let's begin by looking at temperature as the first environmental factor we consider. The range of temperatures for the growth of a given microbial species can be expressed as three cardinal temperatures: minimum temperature, maximum temperature, and optimum temperature. Minimum temperature is the lowest possible temperature that permits a microbe to continue to grow and metabolize products. Its maximum temperature is the highest temperature at which metabolism can proceed and be sustained. And the optimum temperature is a relatively small range between the minimum and maximum, where a fast rate of growth and metabolism is possible. Some microbes have a very narrow cardinal range, while others have a pretty broad one. We have to consider that microbes cannot control their own temperature, and thus they will assume the ambient temperature of their environment. So they must adapt to variations in that environment. Once we get above the maximum temperature, you'll usually see that the enzymes and nucleic acids become inactive or denatured, and thus the cell dies, which is precisely why heat is such a great microbial control agent. Another way that we can express temperature adaptations is whether the organism grows in an optimally cold, moderate, or hot temperature range. We'll consider three basic ranges: the Psychrophile, the Mesophile, and then the Thermophile. You will notice there's psychotroph range here, and then there's extreme thermophile. We'll consider those in some examples, but the main ones will be those three. So we'll begin by looking at the psychrophile. Psychrophiles are microorganisms that have an optimum temperature range between 15 degrees Celsius, but is capable of growth at 0 degrees Celsius. So they're in the low temperature range. True psychrophiles are obligate with respect to cold and cannot grow above 20 degrees C. However, there are some microbes that are considered psychotrophs or facultative psychrophiles. These guys can grow slowly in very cold temperatures. Examples are Staphylococcus aureus and Listeria monocytogenes. These can grow in refrigerated food, and they're a couple of the big causes of foodborne illnesses. Most psychrophiles inhabit lakes, rivers, snowfields, polar ice caps, and deep oceans. The image that you can see below is of red snow, which is a snow algae. It's classically a green algae, but it's dominated by a red pigment at this stage of a life cycle and tends to turn the snow pink. Next, let's look at mesophiles. Mesophiles are organisms that grow at intermediate temperatures. The majority of medically significant microorganisms are mesophiles. Their optimum growth temperature is from about 20 degrees C to 40 degrees C, but they can grow at extremes from 10 degrees C to 50 degrees C. Some of them can survive short exposure to high temperatures. For example, the Clostridium bacteria is a common contaminant of heated or pasteurized foods. And then Giardia cysts can survive for a brief time at these extremes. Most organisms in this group inhabit animals, plants, soil, and water in temperate regions of the world, so subtropical and tropical regions also. Finally, the thermophiles. Thermophiles inhabit soil and waters and are associated with volcanic activity, compost piles, and areas that are directly exposed to the sun. They grow optimally at temperatures greater than 45 degrees Celsius. However, they have a very wide range. Most eukaryotic forms cannot survive above 80 degrees C. However, there are hyperthermophiles that can grow anywhere from 80 degrees C to 120 degrees C. Now, 120 degrees C is sort of understood as the highest temperature at which enzymes can function. There's intense interest in these guys presently, mainly on the part of biotechnology companies. A number of years ago, in the hot springs of Yellowstone, they found a microorganism that they named Thermus aquaticus. It provided us with a heat-stable DNA replicating enzyme that was isolated, which allowed the development of polymerase chain reaction technique. This technique allowed us to take one single strand of DNA and replicate it an unlimited number of times, and thus really thrust the DNA technologies world into the realm that we're in presently. Presently, we're looking at possibilities in waste treatment and bioremediation, as well as heat-dependent industries like baking and brewing and so on and so forth. Now let's look at the effect of gas on microbial growth. Now, the atmosphere's gases that most influence microbial growth are oxygen and carbon dioxide. However, oxygen has the greatest impact. As oxygen enters into cellular reactions, however, it is transformed into several toxic products. Microbes fall into one of these three different categories in the way that they manage these products. There are those that can use oxygen and can detoxify it. There are those that can neither use oxygen nor detoxify it, and those that don't use oxygen, but they can detoxify its toxic byproducts. The destructive metabolic byproducts of oxygen, very often from metabolism, are called free oxide radicals. You can have singlet oxygen with just a single oxygen molecule. It's extremely reactive and essentially wreaks havoc on any living system. There are superoxide ions, which are O2 minuses, hydrogen peroxide, H2O2, or hydroxyl radicals. All these highly negative or electronegative molecules interact with any other charged molecule in the system and wreak havoc. Most cells, though, have developed enzymes that go about scavenging and neutralizing these chemicals. The most common system in aerobic microbes is the superoxide dismutase and catalase system. You can see here that we're taking these two superoxide ions, simply combining them with hydrogen using the enzyme superoxide dismutase, and that results in the production of hydrogen peroxide and oxygen. However, hydrogen peroxide is still toxic. As you know, we use it as an antiseptic. It destroys bacterial cells. So the second step in the game involves these hydrogen peroxide molecules being converted by catalase into simply water and oxygen. So it's a pretty neat system to get rid of all of these nasty little toxins. So there are several general categories of oxygen requirements. This goes for microbes as well as there are aerobes that can use gaseous oxygen in their metabolism, and they possess the enzymes that are necessary to process these toxic oxygen byproducts. And then there are obligate aerobes that simply cannot grow without oxygen. A facultative anaerobe is an aerobe that does not necessarily require oxygen for its metabolism, and it's capable of growth in the absence of it. That is, if oxygen is present, then they utilize the mechanisms of aerobic metabolism. However, if oxygen is not present, they adopt an anaerobic mode, for example, fermentation. A large number of bacterial pathogens actually fall into this facultative anaerobe category. Another category are the microaerophiles. They do not grow at normal atmospheric concentrations of oxygen, but they require a small amount in the metabolism. Most of these guys live in very low oxygen environments like soil, water, and some places in the human body. we'll see that most fungi, protozoans, and many of the bacteria that we study are actually obligate aerobes. Now let's take a look at some of the anaerobes. Anaerobes lack the metabolic enzyme systems for using oxygen in respiration, so they must produce their energy using different metabolic pathways. Then we have strict or obligate anaerobes. These simply cannot tolerate any free oxygen in the immediate environment, and they'll die if they're exposed to it. They live in highly reduced habitats such as deep mud and deep lakes, deep in the oceans and deep soils. The human body also has some small pockets of anaerobic environment, even though most of our body is highly perfused by oxygen. So microhabitats exist where these microbes can colonize and perhaps infect us. For example, around our teeth inside the oral cavity. And then in the large intestine, there's a large amount of anaerobic bacteria. Next, there are aerotolerant anaerobes. They do not utilize oxygen, but they're not going to be killed by exposure to it. So they can survive and grow to a limited extent in its presence. This figure over here on the right shows us some cultures growing on thioglycolate. Thioglycolate is a reducing agent, and when it's placed in these tubes that are exposed to air, then air can make its way in there. But as you get deeper into the thioglycolate, there's less oxygen present. So there's more oxygen towards the top and less oxygen as we move down into the thioglycolate in the tubes. So bacteria that differ in oxygen requirements grow at different levels in these tubes. So we can establish what the oxygen requirement is relatively of each of the organisms growing in here. So all microbes require a small amount of carbon dioxide in their metabolism. However, some will grow best on much higher CO2 concentrations. These are capnophiles. A couple of examples of these are the Neisseria bacteria that cause gonorrhea or meningitis, and then Streptococcus pneumoniae that causes pneumonia. In this figure, we can see a woman working in a chamber that regulates gas concentrations. This sort of chamber is necessary when working in anaerobic conditions. She's outside in an aerobic environment, and the inside is completely anaerobic in this case. In this same environment, we could increase the carbon dioxide levels, so we could culture some of these capnophiles. Microbial growth and survival can also be influenced by pH of the habitat. The majority of organisms live or grow in habitats between 6 and 8. Remember that 7 is completely neutral. That's the pH of pure water. However, extreme acidity has a huge effect on most microorganisms, so 6 to 8 is the preferred range. A few of them, though, live in extremes. These would be the acidophiles or the alkalophiles. They like a very acidic or alkaline environment. Those in the range of 6 to 8 are neutrophiles. On occasion, we'll see some obligate acidophiles or alkalophiles. An example of an obligate acidophile is Euglena mutabilis. This is an algae that grows in very acidic pools between 0 and 1 for the pH. And then there's thermoplasmas. These are an Archaean species that live in hot coal piles. Their preferred pH range is from 1 to 2. If you expose them to a pH of 7, they would lyse because they can't handle that alkalinity. Because many molds and yeasts tolerate moderate acidic environments, they're the most common in spoilage of pickled foods. As we've previously discussed, most microbes live either under hypotonic or isotonic conditions. That is, their surrounding environment has either the same amount of solute as their internal or less solute than their internal. Think of the classic freshwater environment. Osmophiles, however, live in habitats with huge solute concentration. An example of osmophiles are the halophiles. They prefer very high salt concentrations. Some halophiles are obligate, That is, they'll grow optimally in solutions of 25% sodium chloride, but require at least 9% for growth, which is high above the usual environment. There are some facultative halophiles. That means they can take advantage of the salty environment. Some facultative halophiles are resistant to fairly high saline environment. For example, Staphylococcus aureus is one of these species. Because of this resistance, many bacteria and fungi can thrive in these high osmotic environments. Even though we preserve many of our foods with salt or lots of sugar, of course there are some species that actually prefer this, so even that's not quite safe. Now, there are various forms of electromagnetic radiation that hit the Earth: ultraviolet, infrared, and then visible light. And some organisms actually like visible light to use as their energy source. Although there are some non-photosynthetic microbes that tend to even be damaged by the toxic oxygen products that are produced by contact with the light. And the other types of radiation, like ultraviolet and ionizing rays, can also damage most microbes. Another component of the environment we might consider is pressure. Some organisms can handle very extreme pressure. Some deep sea microbes that exist under the hydrostatic pressures ranging from a few times to over 1,000 times the pressure of the atmosphere. Naturally, if we brought them up to the surface of the ocean, their membranes would lyse and they would explode. Now, one thing that all cells require is water. Water is necessary outside the cell in order for transport across the membrane, and of course the internal environment is entirely hydrated. On occasion, as we've explored, some organisms can go into a dormant or dehydrated stage and tolerate this extreme lack of water. But in the end, to grow and metabolize, everything needs water. Let's have a quick concept check. What sort of microbe only grows in the presence of oxygen? Well, our anaerobes, an means not aerobic, so anaerobic cannot live in the presence of oxygen. We'll take that off. A facultative anaerobe, recall, is an anaerobe. It's not aerobic, but it could be. So that doesn't really fall into the only category. An aerobe only exists in the presence of oxygen. And now halophile, well, it could or it could not. It depends on the species. So that's not really an only either. So we'll go with the aerobes. Up until now, we've considered the abiotic influence on microbes, the oxygen content, the pH, so on and so forth. However, maybe even more important than that is the influence and growth of other microorganisms. So most microbes actually live in shared habitats. These interactions can either be beneficial, harmful, or of no particular consequence to the organisms involved. They may be obligatory relationships or not, and they often involve nutritional interactions. Here is a quick breakdown of these various relationships, a great figure for review. They can either be symbiotic or non-symbiotic. Symbiotic means that the organisms live in close nutritional relationships. This relationship is required by either one or both members. In a non-symbiotic relationship, the organisms are free living, and the relationships are not required for survival, but could be advantageous. Let's take a closer look. Under the terms of symbiosis, we've got a situation in which two organisms live together in a close partnership. We could either have mutualism, commensalism, or parasitism. In mutualism, the organisms live in an obligatory but mutually beneficial relationship. Termites are a great example of this. They have a double layer of mutualism, where termites actually have a protozoan inside them. And inside that protozoan is a bacteria. And that bacteria is who actually digests the wood that the termite eats. It's only because of the bacterial enzymes that the termite or the protozoan can get any nutrition from the wood that the termite's eating. That's a pretty neat and complex mutualistic symbiotic relationship. Now, when one member gets a slight advantage over the other, while the other member is neither harmed nor benefited. This is called commensalism, a commensalistic relationship. A great example of commensalism in microbes is seen below in this figure of satellitism. When one member provides nutritional or protective effects that's needed by the other is what satellitism is. So, for example, in this figure, we have a community of Staphylococcus aureus growing down the center. The staph bacteria actually provides a growth factor that's necessary for these Haemophilus influenzae bacteria that are growing in small colonies outside of the major streak of Staphylococcus aureus. So often in satellitism, one member breaks down a substance that would be toxic or inhibitory and thus kill the other member, or they provide some sort of growth factor that helps that bacteria grow where it would not normally be able to grow. There are basically two types of non-symbiotic relationships: synergism and antagonism. Synergism involves an interrelationship between two or more free-living organisms that benefits them but is not absolutely necessary for their survival. Biofilms are a great example of synergism. We've spent some time looking at those already, and we'll take another quick look at them in just a moment. We also may see synergistic infections. This is where we have a combination of organisms producing damage that one organism could not cause on their own. Gum disease and dental caries are a great example. They're both a cause of mixed synergistic infections. Now, antagonism is essentially the opposite. It's an association between free-living species that arises when members of a community compete. One microbe may secrete something into the surrounding environment that inhibits and destroys the other, thus giving itself a larger area in the territory. There are hundreds of naturally occurring antibiotics that we've isolated from bacteria and fungi that have been exhibiting antagonistic relationships. Penicillin is just one of those examples. Now let's take a quick look at biofilms. We're beginning to estimate the significance of biofilms in that they've probably contributed to about 80% of the chronic infections we see today. Below, we see a picture here of a MRSA infection, which is methicillin-resistant Staphylococcus aureus infection. The blue guys in here are the bacteria. The green portions are just fibers of the gauze that it's growing on. Now, most often in biofilms, we have a pioneer species that colonizes the surface, and then those begin to secrete extracellular material which helps keep them on the surface. I'm thinking shower curtain here. In some biofilms, we see association of other species. They can join and they might secrete extra substance into the extracellular matrix. But either way, these organisms are secreting chemicals that allow them to communicate with each other. They essentially monitor the size of their own populations, as well as interact with other species of bacteria in the film. And eventually, we see through this quorum sensing that complex communities are formed. We used to think that bacteria lived relatively independently, didn't really have much community structure or interaction. Well, we've certainly been shown that this is not the case. Bacteria in a biofilm behave way differently than those that are living in planktonic lives or free-floating lives. Even different genes are activated when they're living in a biofilm as opposed to when they're free living. And it's suggested that this quorum sensing, these chemical communications, may be responsible for this alternate gene expression. Turns out that microbes actually exhibit a well-developed capacity to communicate and cooperate in the formation of these communities in biofilms. Pretty amazing stuff. Now, how about the relationships that microbes have with humans? The human body is a rich habitat for symbiotic bacteria, fungi, and a few protozoans. Now, we all are aware that there is a vast number of microbes that normally live on our skin and within our body, all over our mucous membranes. These are the normal microbiota of our human bodies. The relationships can be parasitic, commensal, and synergistic. For example, you're probably aware that thousands of E. coli live in the intestine, and those E. coli actually produce vitamin K for us. Now, there are different species of E. coli that cause some problems for us, but there are many species of E. coli that live in our intestine and keep us alive. Also, another example, lactobacillus live in the vagina. They have a really key role in maintaining an acidic environment. That acidic environment prevents other microorganisms from inhabiting there and creating an infection. So keep in mind that there are literally billions and billions, if not trillions and trillions, of microorganisms living all over our surface, as well as throughout our insides, that are absolutely necessary for our survival. That brings us to the end of this section. I'll see you soon when we study microbial growth.